Without further ado, I'm going to introduce the first of our Frontiers talks, the speaker for our first Frontier talk, Sourav, who is a postdoc in our lab, and he's here to talk to you about therapeutic delivery modes for genome editing.
All right, sounds good. So we've had a really good introduction about what CRISPR is, right? I mean, we are at this point of time when we can make a very precise cut into a genome to do what we want to do, whether that's to basically edit out something that we don't want, that's a mutation, or essentially to introduce something that we do want, which is something that helps a process downstream. And like the popular press is all over CRISPR, right? I mean, we know that. I mean, CRISPR has applications in a lot of things. This is a sort of a leader from nature, and I think we're making progress on each of these elements that I've outlined here, whether that's essentially using CRISPR for creating better xenotransplants, which is here, or even in agriculture to make better crops. But one of the things that I think is going to be critical moving forward is how does it impact human health? And while most of the media or most of the presence is now about making the next super babies, right? I mean, we have these plethora of headlines from all of these popular magazines that talk about that. I'm going to talk about something a little more prosaic and a little more clinically oriented, which is how do you take this technology, which is in a test tube at this point of time, and take it to a patient? And so the important thing to know here is that there are several barriers to taking something that is at the protein level or the gene level and taking it into an organism to basically affect a change. And there are several of them, like you have these organ barriers. Right? I mean, organ barriers have been essentially the rock at which everybody goes to die, or all therapeutics go to die. But at the very basic level, at the very cellular level, essentially you can have a variety of ways in which you can administer your CRISPR components, right? So your CRISPR components are pretty basic. You need the Cas9 protein. You need the guide, which is in the form of an RNA. And if you're doing homology-based repair, you will need a template, right? So there are several ways in which you can introduce it. You can start to introduce your components in the form of a DNA, right? So you can think of essentially virus-based methods like with using AAVs, which I'll focus significantly, like a lot of time of my talk on. Or you could go one step further and say, why do we need to do that? Why don't we actually just put an mRNA or the Cas9 as a protein complex with a guide RNA? And each of them have their pros and cons. And I'll try to touch over the basics of that. And if there is any further questions, we can always do it at the Meet the Editors session. So let's go with the basics of what is becoming one of the industrial or clinical candidates, lead candidates for therapy, which is AAVs, right? So AAVs are essentially viral nanoparticles because they're nanoparticle shaped and can enter and pass through a lot of the barriers that larger particles cannot. So essentially the AAV virus is one of the most simplest things that you can think of. It has a proteinaceous capsid that basically protects a genome that is 4.7 kb long. And what we have done as scientists is basically gut the natural genome of the virus and replace that with the transgene of your choice that is driven by a promoter of your choice, and you have a poly A. And that's as simple as how it gets. So in the case of Cas9, what you need to do is put the Cas as a DNA and run it by the promoter that you want to get expression in the cell type that you want. Fairly straightforward. And there are several advantages of using AAVs for clinical gene therapy or gene augmentation purposes, or even for CRISPR. And I'm mentioning several of them here, but one of them I like to highlight, which is long-term and stable expression of the transgene. So AAVs are extraordinarily stable because they exist as episomes. They don't integrate like lentiviruses, but they exist as episomes. But the issue with that is what is a boon for gene therapy in general is actually something that is not good for CRISPR. Because for gene therapy, what you want is, suppose you're missing a gene, you're missing a protein function, and you want to augment that protein function. You want the protein to get expressed all the time. In CRISPR, you want the protein to get expressed for a very short amount of time, edit the loci that you want, and then just go away. Because if you have it for too long, you're going to get off-target effects. I'm going to talk very briefly about what you can do about it. So let's start with what's being done. So our group is essentially been one of the pioneers for extending the AAV-based Cas9 editing methods. And one of the first attempts was essentially to put the SpCas9 and the guide in 2 different AV vectors. So the problem with SpyCas9 or SpCas9 is essentially the size. SpCas9 is 4.1 kb, and as I told you before, the size limitation of an AAV is around 4.7, 4.8. So you really don't have the flexibility of putting both the SpCas9 being driven by the shortest promoter that you can find, and at the same time having your guide being expressed by a Pol3 promoter. So the workaround that was done in 2014 from our lab was essentially to divide them into 2 AAVs and then to basically co-inject them into the brain of a mouse. And here you can see one of the pioneering examples how using a guide for MECP2, you can basically lower the expression of the MECP2 protein using a guide that is specific for MECP2 in the dentate gyrus of the mouse. Now, a lot of disorders that we're going to face in the clinic are essentially not going to be solved by just targeting one protein, because a lot of these disease components come from multi-gene families. And one of the classical examples is DNMT3s, which have DNMT3A, 3B, and DNMT1, which play sort of compensatory roles in the proper neurological function of a human or of a mouse. And just because you knock down one doesn't mean anything, because the other 2 can essentially come in and sort of compensate that role. So what do? So you'll have to knock down all 3, but the problem with that approach is that SpCas9 does not have an RNase domain of its own. So essentially it has to rely on the host RNase activity or the RNase activity in the host cell to basically cleave it. So what you do is you express each guide from a cassette of its own. And as you can imagine, that's fine for when you're trying to do a 2-vector technique, but you're adding more and more components into this that will essentially keep increasing the size. So here's a good example of how that was done with this U6-driven cassettes, 3 U6-driven cassettes in an array. And here's an example for the DNMT, and you can see that about 1/3 of the cells basically lost all the 3 genes expressed of 1 vector, or I guess 2 vectors in this case. A new tool that was discovered in our lab, CPF1, is a Cas9 alternative. And one of the cool things about CPF1 is that it encodes its own RNAs. And the way we used it is that instead of having 3 different U6s drive their own cassette, you can basically use the RNase activity of this to express all the 3 genes as an array from 1 U6 promoter, and you can have that basically cleave it into its small guide RNA sequences, which are now free to go and actually perform functions for each of these guide RNAs. And as you can see here, a smaller percentage, but a sizable, about 17% of all the cells that we looked at were essentially edited at all the 3 loci using a technique like this. The problem here still is that it's a little too big, right? I mean, yeah, you've decreased the size here, but CPF1 is also big. It's a little smaller than Cas9, but it's really not that small. But why is that a problem, right? Because you're talking about direct brain injections for neurological disorders. The problem is that except a handful of disorders, most neurological disorders are not localized to a small population in the brain, right? I mean, you have the classic example of Parkinson's, right, which is basically dopaminergic neurons in your substantia nigra. Treat that loci, and you potentially can treat essentially the disease. But if you're talking about something like autism, right, I mean, that is pan-brain. You're affecting neurons in every part of the brain. And how are you going to inject? Are you going to do this? And if you think this is a joke, it isn't. So the first AV clinical trial for Canavan's disease used 16 injections into a single brain to get a mild therapeutic effect, not even a pronounced one. So this is really not an exaggeration. So the alternative to that is essentially systemic delivery, right? I mean, why inject into one part of the brain when you can inject into the vasculature and essentially get pan-brain exposure, right? And right now, there's a clinical trial, and as it turns out, there are some serotypes of AAV that are really good at crossing the blood-brain barrier, right? So AAV9, for example, has some limited ability to cross the blood-brain barrier, especially in children who don't have a fully formed blood-brain barrier at the time of injection, right? So you can get across it. And so there's this really good trial that's happening in the, that is being run by Avexis, I believe, with Children's Hospital in Philadelphia, where they're basically using AAV9 to deliver the gene that is deficient in kids who have spinal muscular atrophy. And the preclinical results that are coming out are absolutely fascinating. You're seeing really good event-free progression. So how do you get around this problem? The problem now is that if you use these two vectors and you're doing a systemic delivery, it's fine for the liver where each cell is getting more than one AAV copy. But for a lot of these peripheral loci like the brain, you're essentially going to see one copy of AAV or two copies of AAV. And the fact that you're putting two different AAV particles or two different AAV compositions into the mice basically means that you're not going to have a cell where you have both the guide and the effector, or have both the guide and have the nucleus in them. So the alternative to that is this Cas9 ortholog that was discovered in our lab called SA-Cas9. SA-Cas9 is really small, right? And so the advantage is you can essentially use SA-Cas9 in place of SP-Cas9, have a big honking specific promoter of your choice, have a U6 drive your guide, and still have space to spare. And so here, as a proof of principle, our lab essentially edited the PCSK9 loci, which is involved in hypercholesterolemia, and is essentially involved in one of the key components, key druggable components in the cardiovascular field. And we show that there is about like a huge decrease. I believe it's like 90 to 95% decrease in serum cholesterol after a week of injection. So that was pretty fascinating and really striking. And this paradigm has now been extended quite a bit, right? I mean, it's not limited to just PCSK9 or the brain. I mean, we have had some really pioneering work basically for muscular dystrophy. We had some really pioneering work for some of the other liver disorders. And this is going to get filled up. This space is going to get filled up pretty soon. But one of the problems with this is the thing that I was talking to you about, right? Once you've introduced this, it's going to stay in your cell, and it's going to keep pumping out your SP-Cas9, right? And we know that the longer the SP-Cas9 or any Cas9 is in your cell, the more off-targets you're going to get. It's going to saturate your on-target cutting, and it's going to keep increasing your off-target cutting, right? In principle, at least in vitro. One way around it is to use ESP-Cas9, which is an SP-Cas9 mutant with a high on-target specificity. And so this allows you to get a little bit more of a handle on just restricting your targets to on-target. But what about another way? What about not using viral particles, which is basically the episomal? Why not use something like LNPs, right? So LNPs are great for delivering small or medium-sized RNA particles and have certain in vivo efficacy. They're not the greatest. I mean, we know from the RNAi field that they're not the greatest means to go everywhere, but at least for the liver, they're pretty good, right? So essentially, the idea here is to basically have the Cas9 as an mRNA or as a protein and basically encase it in a liposome or a lipid particle, which basically enters the cell through endocytosis. And so the good things is that there is there's a really pronounced clinical implication for this transient expression, right? Namely the fact that you're not going to get off-target activity. Also, you're not going to get that much toxicity. So you get your improved specificity, and also your nuclease activity is really, really fast, right? Instead of with AV, where you essentially have to wait for your DNA molecule to get transcribed and then translated to have effect. The cons are that while in vivo mRNA delivery by LNPs is getting there, in vivo protein delivery is largely inefficient. In vitro is fine, but in vivo is largely inefficient. And the other problem with LNPs right now is that targeting organs that are not the liver is still challenging. So if you're looking at a liver disorder, look no further. But if you're looking at something like brain or lung or muscle, we'll have to work a little bit on this. So I'd like to round up a little bit. This is my last but one slide. Round up a little bit by talking about what is happening in the clinical trials field. And there are some clinical trials that are starting up. And I'd like to, and you'll see a whole list here. Most of them are ex vivo, which I'll not be talking about, but a couple of them are in vivo, right? So the one from Editas, where they're basically using AAV-mediated delivery of Cas9 to target LCA10, which is a retinal disorder. Their phase 1 trial is planned for 2017, end of 2017, which is pretty close. At the same time, Intellia is going the other route, which is not using AAVs, but using LNPs to target TTR, the liver disorder, and their IND-enabling studies are planned for the second quarter of 2017. So these things are coming up. You'll hear more about them in the news. And if you've not paid attention at all in this talk, this is the only slide you have to pay attention to. Essentially, you have a variety of ways, most importantly a nanoparticle or a viral vector, to deliver by a variety of ways, which is either IV injection, direct injection into the eye, say, or inhalation, like aerosol inhalation, to target a whole range of tissues. And this list will keep on expanding. I'd like to end here, and thanks a lot for your attention.